Okay, well, thank you everyone for coming. It's always good to see a nice big turnout. I think many of you in the room know our speaker, Melissa Haendel, who is a member of our faculty and who now splits her time between here and lovely Corvallis, Oregon State University. And she lives down that way, and so and also has had some great opportunities there. But Melissa has been an excellent contributor to our faculty, teaching and mentoring students and bringing in research projects that a number of the faculty and hopefully over time students and fellows will be involved with, called the Center for Data to Health. Melissa's main research focus is on ontologies and data. Almost anything having to do with biomedical data and ontologies has Melissa's fingerprints on it. So she's very well known nationally, so it's an honor for us to have her on our faculty. Even though she's known around the world for her work and has tons of collaborators, she actually has spent a good part of her life in Oregon. She did her undergraduate at Reed, went off and got a PhD at University of Wisconsin in neuroscience, and then came back and did a postdoc, first at Oregon State, then U of O, and came to OHSU when our library was a recipient of, was it the Vivo project that brought you here?
The Eagle Eye.
Yeah,
Eagle
okay.
Eye.
And then she joined our faculty and has been involved in a number of projects, some of the data science education work that we've done. So it's been a real thrill. I could probably go on and on for like 10 or 20 minutes, but you all came to see her talk. So I will turn it over to Melissa, just reminding anyone who's watching by stream that if you do want to ask a question, tweet with the hashtag DMICEConf, DMICECONF, and we'll try to work those questions in. So take it away, Melissa.
Thanks, Bill. Well, it's obviously a real pleasure for me to be here and talk about some of the things I care about the most with you all. Many of you have participated in various bits and pieces of some of what I'm going to talk about. But yeah, it's really, really a special pleasure for me to be here today. So I'm going to talk about maybe what I think is a little bit less traditional in this context, but I thought it would be really useful, especially for our students, to really think about the impact of how what we do individually or even in our small group projects really can impact how the world reuses our work and how we interact in this greater ecosystem of data. And so I'm going to talk today, the title is The Yellow Brick Road of Open Science, and what are really the barriers to the data that we generate and really in a bucket brigade sort of way, add value to and process and curate and then hand off to the next person for their usage. And when we think about bioinformatics pipelines, there's, kind of an unsung army of biocurators and ontologists that are helping to organize that information that we really forget about the provenance of where that's happening and how that's happening. And so this is really sort of meant to be a little bit of an eye-opener about some of those challenges so that we can prospectively continue to do better. So the cost of genome sequencing has dropped dramatically. And so, of course, the dream is we're going to have precision medicine now, aren't we? But the reality is that in how we go about interpreting it is really the bottleneck and the real expense and the real challenge at this point. So I just wanted to tell a little story. I'm not mostly going to not talk about my actual research today, but rather, but this is just a story from our research. So this is Jessica. Jessica has a rare condition which causes epilepsy and affects her movement, and she has significant developmental delay. This patient had a lot of a battery of genetic testing, and she's a patient in the Genomics England cohort, and we actually solved her case using some of the approaches that I'm going to talk a little bit about. But to solve her case, to understand what her genetic variation is, requires combining her genome with a whole lot of other data that comes from many different places, as well as from model organisms. So we call this challenge the chasm of semantic despair. That's me at the bottom of the chasm. And really what the challenge is that over there on the right, we might have clinical measures for Jessica. We might understand how to measure her developmental delay. We might have laboratory values for her metabolic state. But fundamentally, those clinical data are not very interoperable with our biomedical knowledge that comes from our basic research community of genomics, proteomics, metabolic pathway modeling, and all these kinds of things. And so one of the big goals is to develop ways to make clinical data and basic research data talk to each other so that we can tap into this information for a rare disease patient like Jessica, where we don't actually know anything from the clinical community about her particular disorder. So Jessica actually had her whole exome, actually a whole genome, and she had 6 million variants from the reference genome in her genome. And over 600,000 of those were rare variants. So that's like the corpus of what could be causal for her disease. We filtered that down based on a number of algorithms that predict pathogenicity and changes to proteins, so looking for stops in the protein or things that would change conformational changes. And we get down to 2,800, and then we look at the genetics, and we get down to 67. And using the data from the basic research community, we were able to go from 67, which would take a person's manual interpretation many, many weeks to go through to really figure out what the similarity was to known model organisms or protein interaction partners, to only one by using that data. And this child, so it always makes me cry, is like a happy kid now because she has, she's treated with diet. So it's really the power of that we're after. So this is the world that we live in order to try to make those cases, those wonderful success stories happen. So we have information coming from human disease phenotype resources, which is largely still the only thing that clinicians tap into. But there's this whole wealth of model organism databases and gene expression pathway databases and functional databases and microbiome databases, and they multiply daily in the nucleic acids research special issue. A lot of resources will take data and try to aggregate it and just mash it all into one place and say, okay, well, we can make all this data available. You can search it here. But when you do that, you lose all those special connections that we have that tell us about the knowledge. And what we really want is we want to have things stay linked. We want them to be, we want to understand what the relationship is between variants and a protein interaction partner, or between a disease and a phenotype. So this means that the data brick road is a means, not the destination. And the reason I say that is because how many of you have heard of FAIR data principles? So probably about half. So findable, accessible, interoperable, and reusable. Well, there's a certain amount of it's jump the shark kind of notion with regard to FAIR, because at the end of the day, it's really the R that we care the most about in many of these contexts. So in order to make the R really, make the data really reusable, the three challenges that I'm really going to focus on today are traceable, licensing, and connected, which are fundamental to the kinds of biology that we want to do. So that's really where we're at, just really trying to focus on that interoperability and reusability. And that requires semantic data integration. So just a little bit more about each of these principles. So traceable is all about the evidence, the provenance, and the attribution. Knowing who that bucket brigade member was that did that curation, and can you count on them? Knowing, so a clinician can know where the data came from, so they can know if it's of high value or low value to their interpretation that they're making for their genomic candidate. Licensing, that the licensing for the data can actually, that is clearly stated, comprehensive and non-negotiated, accessible, that it avoids restriction of reuse and avoids restrictions on who may reuse. We're going to come back to this quite a lot, and Robin in our audience has helped, really helped lead this effort. And then connected identifiers, having really good identifier hygiene, data models, ontologies, and concept alignment, and clinical data elements that allow us to cross that chasm of semantic despair. So Identifiers are the invisible bedrock of scientific inquiry. The more complex the question, the more reliance that you need to have on identifier hygiene. So if all we care about is what, we can just have identifiers. But if we want to know something about how many or why or how, we really need to understand what those identifiers mean in different contexts. What's the metadata? What's the data models? What's the context for those links that we want to keep when we integrate data? So equivalence and synonyms are in the eye of the beholder and not always what they seem. So this is an example for the disease known as Ehlers-Danlos syndrome. And it's a complex and poorly understood disease that is known by as many as 40 different synonyms from different database resources. And it's listed in 15 different disease vocabularies, all of which map to each other, right? And so how many of you in this room have combined two data sets that had identifier mappings and they didn't line up in the two directions? Okay, it's like half of you, right? This is like a fundamental problem in biology. Well, if you're a clinician and you want to diagnose your patient with EDS, but your different databases call it something different and have different definitions for it, how in the world are we supposed to solve a rare disease patient's diagnosis if we can't even agree on what the definition is? So this may seem very pedantic, but identification is the foundation to ask very simple questions like, how many people in Oregon have been diagnosed with Ehlers-Danlos syndrome? We don't know the answer to this question, even to this day. What is the collective cost of care of these persons? What are the most cost-effective interventions? Are there any evidence that these patients that are yet to be diagnosed with this syndrome, and how do we reveal those? And Bill's done some interesting work in this area as well, as trying to reveal candidates for a diagnosis just from the underlying data. So the identifier cleaning is a shared pain point and a huge time sink, but there's not really good ways of sharing the outcomes of our identifier cleaning. So for example, let's say I actually went through and conceptually aligned every single Ehlers-Danlos disease representation from every single source. How do I actually share that back so that somebody else can use it? And the reality is there's not a very good way. And I like to joke that the online Mendelian Inheritance in Man database, which has some particular funkinesses with its identifiers, has probably had the same script written to clean the identifiers in the same way by, it must be over 10,000 people by now, right? The same script, right? We're just not good at sharing how we munge that. And unless you can get the source to fix it, which you can't in that case, you're challenged to just go on as we are. So that's a real problem. This is just an example of what we can do when we actually have the right alignments and the right integrations. So this is from a project called the NCATS Biomedical Data Translator, which really tries to align knowledge that's represented in graphs and graph types of operations and analytics and combine them into many different workflows. So it's really developing a workflow language for combining knowledge information sources from hundreds of different resources. And so this is just a snippet of some of that. So if you can take from the Pharos source, you have drug to target associations. So you have disease, drug, target, and you can see some identifiers there, IDG, Haendel, Pharos. We have Reactome has target to pathways, so we can know that a drug target is related to a pathway. In ClinVar, we have disease to variants, so variants are associated with diseases or phenotypes. We have those, but they're using a different terminology. And over here in the database that I work on, the Monarch, we have phenotype to diseases, and we use the human phenotype ontology to associate it with our disease representation, Fanconi anemia. So you can see the challenge, but you can also see how if you start to get really strong alignment of those identifiers, the capability of finding a drug target for a very rare disease starts to become really apparent. So what you need to do with the data, though, depends on the integration approach. So, if we really, download everything, the burdens on the integrator, there's less coordination needed upstream. If we really want to have, the subway map, which is more like the translator, the burden becomes more on the provider, getting those sources to actually have better identifier hygiene, have better change management, have better documentation about their identifiers, how they're provisioned and what they mean in different contexts. So just wanted to talk a little bit about equivalency. So one of the challenges in aligning these knowledge is just fundamentally understanding what is the same in two different places. So, we might have two different equivalent identifiers for the same record that have, and to know that they're the same record is not always trivial. We might have surrogacy or distribution. So there's a European distribution of Medline, for example, versus one here in the US. We have versioning and content evolution. So, conceptually, what we define as a gene today is not at all the same as what it was even 10 years ago. And so there's this conceptual drift. And so, you might find things categorized as genes that really don't meet that current standard anymore. We have related real-world concepts. So chirality, how do two chiral molecules relate to a parent, more generic form that might be represented in a different database. Equivalent real-world concepts that, where we just haven't determined the equivalency yet. We saw this with Fanconi anemia and BRCA-based breast cancer, right? So the same gene turned out to be the underlying causal gene for those two different diseases. And then overlapping records that reference the same identifier. So again, this is this everybody maps to everybody else, but they all do it differently, and those mappings mean something different. When you combine them all together, computationally, you end up with cycles. So you can't perform inference with cycles, and the inability, like the Ehlers-Danlos example, to actually make diagnoses with that example. And if you want to look at more examples, oops. There's a bit.ly there. So because we're actually that nerdy, we wrote a community best practices guide to identifier hygiene. There's also a really fun blog that's been posted on the PLOS Bio about this if you're interested. So identifiers for the 21st century, how to design, provision, and reuse persistent identifiers to maximize the utility and impact of life science data. And it was a 3-year effort from many community members from different contexts. So there's some really good guidance for if you are an identifier minter, please read this paper. I'd love to have your comments too. Okay, so I'm going to move on to a little bit about data, licensing and the ability to reuse and redistribute licensing. So whether the data is actually open or closed for reuse is actually impossible to know until you try. And many of us take data from databases and do stuff with it and do analyses, and then we publish our results. But if you actually want to combine those resources and redistribute them legally, that's a different problem. So when we build our diagnostic tools, we need to redistribute to the clinicians that need the tools. Well, when you go back and look under the hood, many of these resources it's not so easy to tell which ones are open for redistribution and which ones are not. So this really started as part of our work on the Monarch Initiative project, which is the one that builds the diagnostic tools, where we integrate all this data from all these places using ontologies. But what that really means is we have this incredible licensing burden. So over here on the right are just some of the resources that we integrate data from. And these are our institutions. And so for every source that we integrate, there's a pairwise relationship legally that has to be realized in order for us to get legal approval to redistribute. And some of these resources are also distributed, and I'm not even showing that, right? So you can see that the legal burden is just insane. And in fact, in 3 years here at OHSU, I have no data sources licensed in or out. So it's actually really challenging to overcome those burdens. So, and just because we're going to also talk about attribution in a minute, in academia, data licensing is a credit hack and a common pain point. So why do these resources have these restrictive licenses? It's so they can get attribution, so they can tell their funding agency that they're valuable, right? It's a way of sort of making things freely available makes it harder to justify if you don't know who's using your work. It's really an attribution hack. So I love this quote from Ann Carpenter. I would be so happy to never wrangle another legal agreement in my entire life. 7 of the emails preventing inbox 0 involve pestering lawyers just to be able to work with scientists elsewhere. Can we please have a thingy that the entire universe agrees on? So we're working on the thingy. But first, let's talk a little bit about attribution, because that really impacts what the thingy can actually be. So, there's many different definitions of attribution if you go to Wikipedia, but they all essentially, involve, providing attribution to a person or a context for information and the source of that information. So we have this challenge in science of, first author, last author, the contribution statements that you make in your journal. Do you include an author if they provided a paid-for service through a core services facility? I take a very wide view on attribution that everybody should be included. And in some cases, it just takes a lot of effort to get something done. So here's a paper that has over 1,000 authors on it when they, for this Drosophila genomic paper. And so this is the world we live in. Well, what did all these people actually do? The other problem that we know is that in our world, and especially worlds like the work that you all do, is that a lot of the kinds of contributions that we make don't necessarily end up in manuscripts. There's a huge amount of science that never gets published in a manuscript. It gets published on GitHub or clinical instruments or many other kinds of types of things. And this was done some time ago by Nicole Vasilevsky and our colleague Dave Eichmann. And Nicole is also a new faculty here in DMICE. Where we basically got all of the big data to knowledge awardees during a funding period of 3 years. And because NIH couldn't track all the key personnel and give them to us from their database, we emailed them all and found out who were all the key personnel on all of the big data to knowledge grants at NIH and what roles did they have. And then we did a social network analysis just looking at co-authorship. And what we found was that more than 20% of these key personnel were not even writing publications. So 20% of the workforce that's funded as key personnel on the Big Data to Knowledge program are doing other things. They're writing code and other things. So we want to provide ways of attributing those types of contributions. So I wanted to provide a little example, and this is my own personal example, and I have no hard feelings about this example, but it's just the reality of the world and how we can all help each other, especially in the informatics community, to attribute people in the many different contexts that we live in ways that are innovative and really speak to the types of contributions that we all do in informatics. So this is another example from my rare disease community. This was a patient at the Undiagnosed Disease Program at NIH, number 2542. And in this case, we actually did that same kind of analysis where we used all these different biomedical resources to help make a diagnosis for this family. And this involved information, data that was from a mouse that was created by Johannes. And then I curated the information from the patient. So I went through all the patient's medical records and captured semantically all of their phenotypes and all the information about their lab tests and the like. And then Will basically did the analytics where he took information from the basic resources, which included that STEM1 mouse, as well as all of my curated data. And he used that to generate a data set for prioritized variants for this family. And then Tom was the clinician responsible for this patient, and Tom published a paper about this diagnosis. And Tom explicitly credited Will as an author, but not me or Johannes. And so here's the graph, and we're ontologists. And so you can hopefully you can see it's a little small, but there's the publication up there, and the publication is attributed to Tom, who's in orange, and there's an author role and a date. And Tom's also attributed to having made the diagnosis. And so you can see that clinician role and the attribution there. And the diagnosis depended on that data set that was made by Will. So he has a variant analysis role here. And that depends upon two things, Johannes' mouse up there and my curation work over here. But if you have a graph of all of this, you can actually infer that I contributed and Johannes contributed to that paper, right? Even though we're not explicitly credited on the paper. The reality is, though, what do we really care about here? Do we care about the paper? No, we care about the diagnosis. Wouldn't the patient like to know who are all the army of people that actually contributed to their diagnosis? I think that's where we really want to be. So this is actually all the people that contributed to that particular diagnosis. I actually went through our entire workflow system and found every single person that contributed to that diagnosis. Unfortunately, this family was not interested in, due to religious reasons, in sharing this story, so I picked the wrong family for writing a paper about it. But in any case, if the ultimate goal is to reward reusability, we have to clarify the importance and practice of attribution across the translational workforce of all the people that contribute in all the ways that they contribute. Okay, so now that you got the story about attribution, we're going to talk about licensing, because you know now that attribution is really a licensing hack. So, this is a work that was led by Robin and myself and others in our group. And you can find out more information about this work at reusabledata.org, and we have a paper that should be out any day now, and there's a preprint that's up there. But basically, this data shows for 56 different databases that with a very robust set of metrics that, roughly only 20 out of the 56 had clearly stated licenses. You can see that, a little bit larger were at least comprehensive and didn't require negotiation. Finding the license isn't always a given. And then, of course, when you get out here, there are less that don't have restrictions on either the kind of reuse or who may reuse the data. So also, if you look at the license categories, you can see that, there are a fair number that are permissive and don't require legal intervention. But basically, anything that's not that requires some kind of terms or legal intervention or understanding. And there's a lot of, non-standard licenses as well, and every one of those requires a special legal attention. So this has major implications about which data can then be mashed up and redistributed. So over there, up on the right here, you can see a permissively licensed data is like a universal donor. It doesn't get much back from the more restrictively licensed data, but restrictively licensed data can only be combined with permissively licensed data. So you can see that it gets really challenging, and these are all our sources, that when you want to redistribute, you can really only redistribute the slices that meet the most permissive license. And as it turns out, some of the more severe cases are actually from NIH itself, because they have row-level licensing for contributions to places like ClinVar, which has actually recently been changed in part due to our work here. So just a little bit about university licenses. So the technology transfer offices generally do not readily share data or related software under non-commercial licenses. They do not allow rapid data aggregation. They do not protect commercial licensing royalty streams, and they safeguard provenance of research data. But why don't they do this? It's too many data rights holders, universities, preventing the use of data sets, making data integration impractical. Every university wants to protect the IP that we have in our data, which is counterproductive for us mashing it up and redistributing it. So we have this tension. So I wanted to talk about that tension. So how many of you heard of the tragedy of the commons? All right, good. So this is a graph of overfishing of Atlantic cod, and you can see that basically, if we overuse a resource, we don't have it anymore, right? So that's really one of the takeaways there. And then we have the anti-commons, the original robber barons. Over privatization and gauntlets of toll booths meant that trade basically ceased the further down the river you went. And this is actually one of the castles that took tolls on those trade routes back in the day. So beautiful castle. So here's the actual story. So if we think about, so people say you must not be an open science enthusiast if you don't want to make everything Creative Commons type 0, which basically means free for anybody under any circumstances. And that would be fully open access over here. And so that's not really true. On the other hand, what technology transfer offices want is over here, and that means we have to reinvent the wheel every single time to get the data because we can't actually combine things. What we really want to be the zone of cooperative and market-based solutions is really over here, and this is what they call the wealth of the commons. So we need solutions that provide the attribution, but also provide the ability to freely redistribute under those licensing terms. And I actually think that we don't actually have a good license for data to date. And this is just an example of what some of those standard licenses are from Creative Commons. So you can see CC 0 lets you copy and publish, use commercially, modify, adapt, change the license, publish without attribution. And as you get down here to the more restrictive ones, you have attribution, non-commercial, no derivatives. And you'd think, okay, maybe it's really simple. We'll just go with CC BY, and maybe that solves the problem. But the problem is that CC BY just says you just have to have attribution, But there's two problems with that. One is that the attribution has to be computable. So if I've got data that's been mashed up and then I've mashed it up again, that license has to flow with the data. So but attribution is an additional requirement computationally that I have to track. And when the contributions are made by individuals on every edge in the graph, that's a whole lot of data that has to come along in terms of licenses. So there's a burden of having CC BY. But if we go back to CC0, we're back over here where we're going to have overfishing, right? So the other thing is that it's not always so easy. So on some of our work, for example, on one of our ontologies, we have a custom license that has a CC BY no derivative type license. And the reason is because we don't want people to take our data standard and make derivations of it and not give it back to the core because it's used for rare disease diagnosis. So if companies go and take it and make their own derivatives and don't give back to the core, we're actually less able to solve more patients' cases worldwide. And so it's an ethical reason for actually having no derivations. But that's, of course, not considered open science either. So anyway, I'm probably running out of time. How am I doing for time? I'm fine? OK, great. So why not CC0 then? So pros is that anything is better than gridlock. It's very quick and easy to implement. It's digital data in the commons. It's inexhaustible, cannot be overused. Currently supported by the Creative Commons initiative, so we have a standard license for it, and NIH tentatively supports it for biomedical data. The cons are that it doesn't address software licensing. So data without related software often doesn't have the same value or is otherwise inaccessible. And the Bayh-Dole Act implicitly protects software against patent rights, but not data. We also lose provenance on the research data. As I mentioned, that provenance and attribution is really important for knowing how to justify whether or not that data is important for the clinical use that you have. So if we go with CC0, we're going to lose a lot of that really important extra information that comes along with the attribution and the provenance. We abolish commercial royalty streams on data sets. We have a real sustainability issue. If everybody makes things CC0, that means that our taxpayer dollars have to continue to fund those resources through NIH and other funding means, right? So there's not really a built-in sustainability vehicle in that context. And it doesn't solve the problem of various rights holders retaining their copyrights in their database submissions. So we have to, NIH can say, ClinVar is CC0, but the actual row-level contributions still say those copyright owners are the rows of the submission. So they have just changed that, but that's a problem still in a number of other databases. So maybe we need a new Goldilocks Commons license for data. And I also have this idea around a data trust, and I'd love to hear what people's thoughts are on this. So a new universal license would be similar to a CC BY, so attribution, non-commercial, share-alike license with no legal negotiation required. And all universities would join a club to have this, similar to what we've done for the universal MBTA, which is so that we can transfer materials between universities. So it used to be that we had a similar problem for when I wanted to send a mouse or an antibody from one institution to another, because we would have to get legal intervention to make an agreement, because that's IP from that institution. And so now, because of the universal MBTA, all the universities have joined the club. There's no legal intervention now required. So we need something like that for data. And we have a committee that's started for the Association of University Technology Managers, Autumn, that's going to be working on this problem. So the proposal I have is that we actually have something called a data trust, which could include some kind of nonprofit entity for organizations that join the club, and that the revenue would be distributed. So we would have the commercial organizations could pay some kind of licensing fee, or there could be some per use, and you could come up with a number of different models where you would get some kind of revenue from commercial users, but make it free for research and non-commercial users. And that revenue would be redistributed amongst the participants in the trust to provide that sustainability vehicle. So we have our integrated data. Commercial entities come to us all the time and say, we would like to license your aggregated, integrated corpus of data, and we say, well, you've got to contact these 100 different resources to make your own legal agreements because we don't have any way of doing that. So this would sort of, help that, the value of that data, especially in its integrated form, be a licensable entity that could provide sustainability for all those resources, which would then help with the attribution and provenance because people would actually still have all the attribution and provenance in that context. And then funding agencies would promote the idea of the organizations joining the trust. And so this would, this kind of a trust could really solve the problem also of how we actually evaluate a data resource. So not all data resources are created equal. They come and go. They have different levels of robustness. They, some graduate students' project was really great, but then they graduated and they moved on and it wasn't maintained. How do we sort of provide the value proposition? And so whatever the trust model would be for commercial licensing, it would actually incentivize participants to have their data resources be as robust as possible, which would solve that way that we share our knowledge about how we integrate data and our identifier best practices, and all those kinds of things would have to improve in order to get a larger chunk of your licensing revenue. So with all of that, I just wanted to thank all the people that did the work here. I've probably missed lots and lots of people, but especially Robin for the data licensing work, all of my colleagues in the Monarch Initiative, but especially Nicole and Matt, who've done a lot of the identifier cleaning, along with Julie and Kent. And lots of different projects that we work on really rely on good identifiers and hopefully soon better licensing for our data. So thanks very much.
Use the microphones so the talk is both recorded and people who are streaming and can listen to it. But since I have the microphone, I will ask a question. I was recently involved in a pretty deep discussion about, and I noticed it's really missing from your discussion, although you gave a very nice comprehensive overview, but is what are the role in the medical context of patients? What's the role of them and their data? And, there's various ideas that are kicked around that, for example, and especially outside of medicine and public health, that people should be able to sell their data. And that could create a wrinkle for those of us who want to do biomedical research with patient data. So I could go on, but I wonder if you have any thoughts on kind of where that might all fit in, and could there be impacts if there isn't some sort of common use that could be done, could be legislated or whatever, regulated around patient data?
So there's a lot of things to talk about with that. So the first one is just interoperability for patients, right? So which is kind of even just the first step. And so one thing that we've done is, so like our terminology that we use for clinical phenotyping, we've translated it into many different languages for global use, but we've also translated it into laypersonese so that the patient can actually use the same identifier for the thing that they have in a way that they can recognize. So that's, I think, thinking about how patients just interact with this ecosystem is one question, and that's one way that we absolutely have to have in order for that to work, is that the same content should be able to have different views for different contexts. So patients need to see things in a different way than a basic science researcher or a clinician, but we need to cross that chasm of semantic despair, not for just basic science and clinical science, but also for the patient. So that's one thing. The second thing, I think, is just as a patient own their data, and we already are challenged even just with data use agreements and permissions and consent not being very computable. And so, I like to joke that a couple years ago I was at the doctor's and they're, please sign this form saying that we're not going to share your data. I'm like, okay, I want to sign the form where you share all my data. And they're like, well, we can't treat you because we don't have one of those forms. There's no way for me to sign a form that says I actually want to share all of my medical data. Anyway, so we don't really actually give the authority to the patient to make those decisions for themselves right now. They walk into the office and the assumption is that we should protect everything. And then even when we do, we say, okay, for research purposes, but what does that actually mean? And that's where I think the patients over time need to have more, we need to have more control over, similar to how you are on like Facebook, for example, which people, your friends of friends, the public can see things. We need to have kind of different layers of permissions and consent that can match the different kinds of computational usage. And those kinds of permissions and consent, if they were more computable, could be aligned with financial sustainability kinds of solutions, so that, similar to Johannes's mouse, so wouldn't Johannes like to know that his actually good phenotyping of his mouse led to the diagnosis of a patient? But what if he also got back, $10 every time every diagnosis was made using that mouse? Well, it's the same thing for a patient. If they're contributing their data, maybe there's a data trust, that kind of function that can work for them as well, so that if they contribute their data to research, it's clear what kind of research. But then if it's used and it's actually useful, there's some kind of benefit to them. And sometimes it's just a social benefit, not a financial one. I'm sure Johannes couldn't care less if he gets $10 for having made this diagnosis, but he'd sure like to know that there was a diagnosis made. So most patients feel that way too. So I don't know if other people have answers to that question. I think it's a really important one.
I was wondering, on your side, I think it was the second to last slide, you had, your trust idea, and I'm like sitting here trying to think of what would be like the negatives of that, and what have the doubters sort of come to you saying, oh, that won't work? Like, what are the greatest, challenges, do you think, to actually making that a reality?
So I think there's a trust issue. And, it doesn't work if only two groups decide to join the trust. Like, we really got to get everybody to try to work together. And that's never going to happen until the funding agencies get on board with this idea. Because if the funding agencies still put people through the same, it's one thing to fund, hypothesis-driven research or clinical trials, but to fund a data resource production source, we should have different ways of evaluating them and different ways of supporting them that are more, built in to incentivize their quality production and evolution over time. And those funding strategies right now they just treat them the same as any hypothesis-driven science. And until that sort of general notion, and it's changing. I've been emailing lots of people at NIH and meeting with them for many years now, and it's starting to sink in, and so have many others. So I think it is going to change, but we have to have the partnership of the funding agencies and the providers to all at least pilot something, and we could start small, and just get 5 or 10 resources to just try it together, and then show that it works, and then we could grow it from there. But I think that the resources themselves will never do anything without the stick from the funding agencies, and then they don't trust that their best interests are in mind, because they're so used to being afraid of losing their funding, through this not very good process of review that these types of resources go through. So I think it's a little bit of a catch-22, because we need the community to tell the funding agencies that this is how it should be, but we also need the funding agencies to tell the resources that this is how it's going to be, and try to coordinate that cycle. There's no cons to it. It's just who's going to get paid to do that. I already spend like 1 day a week on my data licensing problems, so we just need somebody at NIH to kind of break the catch-22, I think. Yeah.
I think two other things that are related to that is, one, which you touched on, the work that goes into taking a data set from the point where the person who created it can use it to the point where other people can reuse it is it's an extensive amount of work.
Yep.
And it can be daunting, and it's generally kind of unrewarded work. And obviously, like, this exact proposal would be in some ways generating the benefit for doing that kind of work, but it's non-trivial, and that's part of the problem. And then the other thing related to the patient issue is issues of privacy. And anyone who is working with or creating a data set based on patient data always has these concerns about patient privacy and what's permittable and what should be done. And you know, you were talking about the fact that this consent process needs to be computable. And I know that there's also discussion that the idea of informed consent at its core has not kept up pace with the way that biomedical science has developed. And that something really has to change so that there is a better understanding between researchers and providers and patients about what they are actually agreeing to. And that would have the added benefit of potentially clearing up some of these data sharing and reuse issues. But it's pretty gnarly right now to try and wade through that as a researcher.
Yeah, and so then it puts the fear of God in you, and you do nothing because you're too afraid to, as we all should be, but it's sort of a blocker.
I've had literal panic attacks about like privacy issues.
Yeah, me too. So do you guys feel empowered to make good identifiers and attribute all the people that touch them?
Actually, let me ask you a question about that. I mean, you described it a little bit, but you know, I too, you know, sometimes come across those, you know, papers that have 1,000 authors, and you know, I really wonder, you know, how many people who are on that author list actually wrote a single word in this paper. And so, you know, how can we, you know, especially for academia, I mean, you know, for us to advance and get credit, or as you say, to get credit in making diagnosis, how do you envision that will happen, that there are ways to attribute contributions to these sorts of projects,
well,
sorts of research?
That is something that we're working on. Couple different things. You know, I think one is we actually worked really hard not that long ago. Some people complained about this, but to get the NIH BioSketch format changed, so that so you know, when it went from 3 pages to 5 pages, and everybody complained because it took them a whole day to redo their BioSketch. Well, you can blame that partly on me, but part of the reason was so that we could actually put non-traditional types of contributions on there. So it doesn't all have to be published literature, but it could be a code base or a clinical instrument, or diagnosis of a new family of genes, or you know, of diseases. So you know, I would urge all of you in that context, or on your CV, to list all the kinds of contributions that you make to science. Story I'll tell, I was at a rare disease foundation meeting, which was 1/3 patients, 1/3 clinical physicians in that field, and 1/3 basic science researchers. And you know, we were told that we were not allowed to take photos or tweet of any kind at this meeting, that this was supposed to be a, quote, safe space for us to work on this rare disease together. And I just stood up at the town hall at the end, and I said, I'm really surprised. This is the first time I had been to this particular meeting. I'm really surprised that you guys aren't, like, you know, announcing to the world what you're all doing and getting the world to work on your rare disease. Why aren't, you know, couldn't we incentivize many more people to help solve the problems that you have if you know, we're sharing your information about what you're doing, sharing your data, sharing your technologies. And one of the more, you know, senior people in the community stood up and said, but if we do that, our graduate students and postdocs won't be able to get that cell paper that will get them a job at Harvard. And I said, you know, I think that we have the potential to change this one CV at a time, right? So if you at Harvard only hire people whose CVs have the things on it that show that diverse kinds of contribution, that's how we're going to change it. So we all have to go back, so your homework is to go back and fix your CV, your BioSketch, and make sure there's lots of stuff on there that is not a paper, so that the world can know that you do other things besides write papers, because I know you all do because you're in the informatics department. So, you know, Zenodo is your friend. Publish things, get persistent identifiers for them. You know, and for anyone who ends up being on a hiring committee, look for that and value those kinds of diverse contributions. We also, that's part one. Part two is that we also are working on a contribution role ontology, as you might imagine. And that ontology will have a little data model associated which allows you to record the kinds of contributions to any kind of entity. So if you have, we can record contributions to an ontology, we can record contributions to a clinical best practice guide, we can record contributions to, a computational package. And we can say what were the roles that the people had in that. And that would actually allow me to have said, what were the roles, if I go all the way back to these guys, what were all the roles that, that these people actually had in that diagnosis. I would be able to do that. And I could also associate that little file with the paper. So even though they weren't authors on the paper, so they didn't write a word of the paper, that doesn't mean they didn't contribute to the science, right? And so right now, the acknowledgments section of a paper is really a proxy for these types of more distant contributions, but many of which are pivotal. And this type of structure would actually let us include everybody that contributed. So yeah, I thought about it a little bit too much, probably. Yeah, go ahead.
So is there any conversation among the International Committee of Medical Journal Editors or National Library of Medicine for PubMed about actually having the acknowledgments field be a parsable, discoverable field in a standardized way so that when people are Googling you to, find your, publications in PubMed, that they find these other kind of activities, because otherwise it's just, they don't have very many publications in PubMed, and they don't even get to the level of looking at your biosketch necessarily.
Yeah, absolutely. So there are conversations going on, and I know Robin's had some of these conversations too. We've actually mined all the acknowledgments of all the papers that are in Medline, and we have them all in one big database, and we've associated all of those people's names that we could with people with ORCIDs or other authors that are in Medline. So we actually now know who all those people were. It's a little harder to figure out what they actually did, but we do have just sentence parsing of those phrases. So it says, Melissa made a mouse, right? And so mouse would be in the same sentence as Melissa. So we have those phrases parsed, so we can kind of get a sense of that. We've done some classification of that. And there's been some other published studies that have done some really interesting classification of mining of acknowledgments as well. I did have a conversation with NLM recently about this exact problem, and also the notion that we not just for this purpose, but for many purposes, how can the community build tools on top of Medline? Because Medline is really a quality collection of journals that provide quality science. We should have the ability to build lots of different kinds of tools on top of the literature and share those, which kind of gets back to that sharing problem, instead of either relying solely on NLM to provide them or running over here and finding somebody's GitHub account and running your own thing, to create more of like a tool shed around Medline. And so this is where the main conversation has been, is to try to think about how do we create a community-driven tool shed for functions like this to live on top of Medline. So, and if anybody wants to participate in this effort, I would love some partners, so let me know. Yeah. Any other questions or thoughts? We could go around the room and hear somebody's identifier pain story. Anybody want to share the last thought? Good identifier story?
Well actually, you mentioned orchids, and I was just going to encourage everyone, if you haven't already done so, to and get one. It's a simple process, and a lot of journals, I've submitted to some journals where that's how you identify yourself. So participating, especially as you become researchers in these sorts of activities, is important too.
And if you need any help with these, contact the library.
Yes. Robin Champeau in the library.
Your go-to person.
1 or 2 floors below us can do that. Well, okay. We're actually just about out of time, so I want to thank Melissa for talking. I'm looking forward myself to visiting Oregon State next week.
I know, it's so cool. We're doing like reciprocal visits this 2-week period.
Thank you, and you'll stick around if there's any questions.
Okay, thanks everybody.